All right, yo, right now I can actually project from my iPad, so hopefully this works out. All right. All right, so good morning, everyone. So today officially is a seminar presentation. So what I did was to provide you with articles that dig into the topic of our focus, which has been this idea of balancing metals. And so we discussed this from the context last time of Alzheimer's disease, which is a disease that to some extent is believed to be metal dependent, if not necessarily metal caused. That's something that's still very much debated, the actual causation of that particular condition. So we're going to continue on looking at a few different examples where we're looking at this idea of metals in terms of the loss of homeostasis, and also if we have time, I don't think we'll be able to complete this particular lecture until the next period. We'll look at how a metal that's not necessarily essential can give us insight into this loss of regulation of a pathway, biological pathway. All right, so this is what we discussed last time in terms of our introduction about metal ion homeostasis and regulation. And what I want to do is really zoom into this component of it. And so we're going to look at another example where listed as B here, where we talk about this idea of losing that internal homeostatic control and how the body responds in a manner that is aberrant, but also in a disease state. So I gave you all this article to read, and we're going to look into it. Really read this article, which talks about the use of a small molecule, a chelator, and how it could potentially be used for this idea of redistributing iron in a condition where there's a ferroporin deficiency. So a ferroporin deficiency can lead to a variety of different types of diseases. And so we talked a little bit about this idea of chelators potentially being used as a way of redistributing metals. And so here is an example where they really looked into this possibility with a particular chelator. All right, so in our discussion, I want to show you this figure. I used it a lot from my own discussion of the research that I do, but I just want to draw our attention to the importance of this particular transporter, ferroporin. And so here, I tend to use it as a way of contrasting what goes on in the healthy cell versus what goes on in a cancer cell. And when you see this idea that in a healthy cell, we have this balance of the amount of iron that's going in and the amount of iron that's leaving. So you have this control of the iron. You have an optimal level and you're not reaching an overload. But then what you see in a cancerous cell is that that loss of homeostatic control is actually desired by these types of cells because they want this overload of iron, as they do depend on iron for a progression. So this idea of having a lower expression of that transporter, the ferroporin, which controls when iron gets out, is something that when we're talking about a condition where we have too much iron building up, can really lead to a lot of toxicity. So that's going to be the focus of this particular article here. So let's talk a little bit more about the properties of the ferroporin transporter. So it is a 12 transmembrane helix protein. And so here I'm just representing to you the structures that correspond to this particular protein. Those are the transmembranes. And here's just a perspective of it with all the apohelical structure that they have. It is known to be the only iron exporter in mammals. And it plays a very key role in iron recycling. So there's something that is essential for controlling, again, the amount of iron, the concentration of iron that we have. And so here we have this perspective of the function that it plays, where essentially you have that particular protein serving as a transporter. So here's just this idea of moving iron from the cellular environment, in this case, to the extracellular environment, to the bloodstream, especially when you have this, let's say you're starting to fall right below the optimal level of iron in the body. And so there's this, it senses that there's a decrease in that iron, then you have mobilization from ferritin, and then this iron will get exported out through this particular transporter. But then when you have a situation where there's simply too much iron, you have this other protein or this hormone that helps to regulate the control of how much of that particular protein is accessible. So that interaction of the hepcidin interacting or coming into contact with ferroporin is important, and that interaction leads to internalization and basically a decrease in the amount of ferroporin that's available. All right, so it is regulated post-transitioning. So what that means is that after it's expressed, then that is when that particular hormone regulates the amount of how much it is actually expressed. So loss of its functions due to genetic deficiencies or dysregulation of that hormone, hepcidin, can cause iron misdistribution, and that can lead to particular iron accumulation in the liver and its deficiency in bone marrow. So its deficiency in bone marrow is important because then that can lead to a decrease in the amount of red blood cells that can be produced. All right, so ferroportin disease basically has to do with when you have mutations that are functional mutations, essentially removing the capacity of ferroportin being able to serve that function of transporting iron. And so that's why here it's caused by loss of function mutations. And so here are some of the major amino acids that when they get mutated, okay, they can lead to a loss of the function. So some of the ramifications of this is mild anemia and then in liver iron overload. All right, so currently the treatments for this particular condition are suboptimal. What I mean by suboptimal is that they're not necessarily targeting the causation of it. So you have one form, which is venesection, that's essentially like drawing blood. So you're trying to minimize the amount of red blood cells because you're trying to minimize this iron overload, or the use of iron chelators. And the thing is that the lack of specificity of certain of these iron chelators contributes to this. And also the inability of certain iron chelators to potentially participate in redistribution of that iron. All right. So here, one of the real focus is trying to look at the use of a small molecule to potentially engage in metal distribution. And so this is work that predates the article that you're actually were assigned to look at. So here is this the chelator, the hinochitiol, and it is actually a natural product. And so here I'm showing you its coordination to iron. So here's where you have the sites of coordination. So the bidentate ligand, when it saturates the coordination sites of the iron itself, you have 3 of the molecules coordinated to that metal, and that's believed to be the physiologically relevant form of the iron complexation. And to the right here, what we have is a cyclophotomegram that was collected at a pH of 7.2. So the importance of this is to give us an understanding of the potential of the iron centers. We're looking here at the iron 3 getting reduced to iron 2. So we're looking at the potential of that that particular complex to be able to flux between iron 3 and iron 2. So let's look at a few different features about this. So this particular chelator and its possibility of engaging metal redistribution was found from a type of complementation experiment. So what was done was that a series of iron chelators were studied. So this particular hinochitiol and some of the more classical chelators, synthetically made chelators, were explored. And the idea was to understand, its possibility of trying to improve the metal transport in the yeast, Saccharomyces cerevisiae, because that particular yeast that they were studying was missing the iron transporting complex, the FET-FTR1. And so of all the chelators that were assessed, this was the only molecule that actually had the capacity to restore the transporting of the iron itself. So that was the study in JACC in 2015. So a few of the things that they characterized was the affinity for iron. So here we have the affinity for both iron 2 and iron 3 when it's complex with 3 molecules of the ligand. So here we have our beta constant. And you can see that clearly iron 3 has a much higher affinity, but still this ligand has a significant amount of affinity for iron 2. And so with the cyclophotometry that's collected there, we can see that the iron 3 is likely to get reduced if we're talking about the reducing environment of cells to iron 2. So that can be a redox active metal center when complex with that ligand. So they have that represented here with the reduction potential of negative 361 millivoltage versus NHE. So that's within the biological range. Another really important thing is that it has a very high binding capacity. But is that capacity labile? So that experiment in 2015 suggested that it would be. If it does have the potential to restore the transport of the metal, is it something that can be labile? And so this experiment was done here, where you had iron bound to the ligand. So I'm not sure why I abbreviated H. I meant to abbreviate this, but I can correct the slide. Anyways, so you have one of the iron 56. And so what they did was an exchange experiment with iron that was the isotope 55. And what they were able to observe was that the isotopes were able to exchange. So this is one of those examples of a high affinity complex, so very stable, but also labile. So this is something that was able to exchange about 20% within 10 minutes. So within the biological context, that is considered something that's fairly labile. So that's an important feature in terms of the possibility of not only being able to grab onto the metal, but to also be able to exchange it. So additional things that were observed. So it has the capacity to perform protein transporter-like ion mobilization in a site and direction selection manner by regulating the transmembrane ion gradients that accumulate where protein ion transporters are missing. But the question is, and that's what really leads to some of the studies in this particular article that I had you to sign, is like, what's governing that redistribution? So it was observed that the ligand has the capacity to increase gut iron absorption in the divalent metal transporter. So that's a transporter we've talked about a bit in terms of participating in the iron endocytosis within our cells. Like, it has that capacity of increasing iron absorption in these animal models. And in fact, this model here is the one that is really one of the principal ones in the article that we're going to focus on, which is the flat iron mice model or mouse model. That model is a representation of ferroportin deficiency or the ferroportin disease. Also observed was that it had the capacity to induce hemoglobinization. Okay, so what does that mean? So as part of being able to participate in the regeneration of red blood cells, it has the capacity to increase the amount of hemoglobin that's available. And that was observed in that animal model, zebrafish. Okay. All right, so with that background at hand, then, what they wanted to focus on in this article was to look at the possibility of this ligand potentially participating in the human body. So the question is, and so this is basically their figure in which they tried to propose their own hypothesis. So they wanted to understand the following: to explore an alternative approach to the treatment of ferroportin disease by using a small molecule surrogate for the FPN1 that would redistribute iron from liver macrophages. Macrophages are a type of white blood cells. I try to define certain things just in case we're unfamiliar with them. I myself am unfamiliar with any of these terms. To bone marrow to reestablish this iron homeostasis. All right. So this figure on the right gives us this perspective of what they're trying to achieve. And so actually, this is really a nice figure. So the idea is that there's a deficiency of the ferroportin in the liver macrophages. Okay, so the idea is that, can this ligand have the capacity to remove the iron from the liver cell and then be able to relocate that iron in order to restore the formation of red blood cells generated in bone marrow, and then give you those mature erythrocytes that can then lead to the hemoglobin. So try to reestablish that hemoglobinization. So they wanted to explore this in a few different contexts. The possibility of moving iron that's overloaded into the liver macrophages, one thing. Then they wanted to look at this interaction is the movement after it complexes that iron. Can it actually deliver to transferrin, and does transferrin participate in the delivery of the metal to the bone marrow? All right, so here is their overall approach. So first, they wanted to examine the capacity of pinoculio to induce hemoglobinization in the ferroportin disease mice model, so the flat iron mice. So essentially, they wanted to look at, can they mobilize iron from the liver to the red blood cell? So that was the first thing that they did. The next, they wanted to examine the capacity of the ligand to mobilize iron out of ferroportin 1 deficient macrophages. So can it actually have that capacity of moving it out of these white blood cells? And then to assess whether the ligand can induce hemoglobinization in a serum transferrin dependent manner. And then finally, with using this context then to go to the human level. So not working with humans directly, but working with a clinically relevant system, so in an ex vivo fashion, to examine the potential for the iron redistribution by the actual ligand. All right, so this is the mouse model that they use, and that is the lead animal model for ferroportin disease. And so it features this mutation, this custody for arginine loss of function ferroportin 1 mutation. And so the physiological impact of this is the observation of an iron buildup in the liver. So that's classical of the actual disease. So it mimics that. There's also a reduced level of iron saturation, so the serum transferrin saturation. So there's a decreased amount of iron that's accessible in the blood. And also this particular animal suffers from mild anemia. So it mimics many of the characterizations of the actual disease condition. All right. So towards the first objective or the approach. So to examine the capacity of the ligand to induce hemoglobinization in the mice model by mobilizing iron from liver to red blood cells. So what they decided to do was perform an acute IP administration. So IP administration is an administration via the gut. And they did this using various dosages of the henochitiol, so from 1 to 50 milligrams per kilogram of animal. So this is considered an acute administration because it's something that they assess after 4 hours. Okay, so what is the immediate effect or relatively immediate effect? All right, so what are we seeing here? So we have a profile of the amount of non-heme iron. Okay, when you're talking about non-heme iron, typically you're thinking about something that's not bound, obviously, to the heme group. Potentially could be a measure of labile iron. And you're looking at this as gauged within this mouse model at different dosages. And what are we seeing, Brenda? What do you see in this level of the from 0 dosage all the way to 50 milligrams per kilograms? What's happening to the iron? It's decreasing, okay? And according to what they're writing here, you see these stars? What do those stars mean, Julie? Good. It has to do with what they call statistical significance, which is actually a very abused terminology. Many people use it incorrectly, and they love to put many, many stars, because many stars means very, very significant, and oftentimes it's actually exaggerated. But whenever you see this, it implies that there is a significant difference from whatever they're comparing. So you see how they have from here to here. It's telling you that there's a significant difference from the 0 point to the 0 dosage to the 50. Okay. And so another observation. So compared to wild type. Okay. So this is a non-disease mouse model. Compared to wild type, Javier, 50 mg per kilogram in the disease mouse versus the wild type. What do you see? Any difference? All right, so Javier says quite similar, and that's correct. So the idea is that this administration was able to get to basically wild type level. All right. And so when you assess total iron concentration, so this is going to be everything that's accessible, we see the very same trend that Brenda pointed out, decrease, and Javier pointed out this. Oscar, what are all these dots here? What are all those dots?
I mean, ¿qué significa el punto de arriba? La dosis que se está administrando.
No, it means all the times that they actually administer that dose. So you'll see a lot of a tendency to move towards that, actually showing the real data. Because sometimes the bar graphs can be misleading. So sometimes the journals will tell you, put in all your real data so we can see the spread of the actual numbers. Okay, so then they looked at iron as assessed in other organs. So they looked at the spleen, they looked at the duodenum, okay, that's part of the gut. All right, and what do we see here? All right, Carmen, what do you see here? Yeah, well, it looks like it's a little bit higher than wild type, but the key thing is that there's no difference across the dosages. So it doesn't look like that ligand is affecting the iron there. So it seems to be site selective, right? So it's organ selective. And so something is generally true of the other tissues, probably within reason. Because if you look here, there's a trend towards a decrease, but not statistically significant according to them. All right. And then we look at the profile to other metals, right? Because this is a ligand that also has other metal binding capability. And it turns out, so there's a lot of figures here that they looked at manganese, zinc, and copper in the liver, the spleen, the duodenum, and they don't see any changes in the levels of those metals. So metal selective apparently within this animal model. All right. So then next they did a chronic IP administration. So chronic means longer term. So 10 milligrams per kilogram of the ligand daily for 1 week. All right. And so what was observed? So a very similar behavior as observed for the acute was observed for the chronic, where you have a decrease in the liver, and really not any significant decrease within the spleen and the duodenum. So still seems to retain that same pattern. All right. So when we look at other components of this, so they're able to establish a movement of a decrease of the iron that's in the liver. But then what are the other features that they see? So do they see an increase in the iron that's in the blood, right? Because that's the idea. You're moving out of the liver, are you now able to recirculate into the circulatory system? And are we also seeing this corollary to iron binding to transferrin? And so what do we see? So according to this, you do have an increase in the amount of iron that's available in the blood of the animal. Okay, so this was, again, the chronic administration. You also have an increase in the amount of iron that's bound to transferrin. And interestingly enough, no effect on the ferritin. There is some amount of ferritin that's in our circulatory system. So remember, that is our storage protein, our cells, but there is some amount that's available in our cell, and that's actually not getting affected. All right. So then what else do we observe? So now we have iron moving out of the liver. We also have iron being bound to transferrin, more iron in our blood. Now, do we actually see the formation of red blood cells? So that is what what that term is, the hematocrit. That is basically a measure of the red blood cells that are being generated. And then also corollary to that, do we see an increase in the amount of hemoglobin available? And so what we observe is that, yes, we have that pattern, increase in the hematocrit, and then the hemoglobin. So you do have an effective hemoglobinization in that particular animal model. All right. So now the next thing, to examine the capacity of the ligand to mobilize iron out of the ferroporin 1 deficient macrophages. All right. So now we moved away from the animal model. We're going to look specifically at these liver cells. Okay. And the liver cells of that are the macrophages, so these white blood cells. And so they refer to them as the Kupffer cells. Okay. So the first thing they did was they used these raw 264.7 macrophage cells to generate an FPN1 knockout macrophage. All right, knockout. Brenda, what does knockout mean? All right, Javier, what's knockout mean? I know you guys Google right away. As soon as you don't know something, know you that's what you're doing. That's what I certainly do. Sorry, no shame here. Javier, what does knockout? Okay, knockout means that you knocked out the gene. In other words, that will not be expressed. Okay. All right. So we have a knockout of the ferroporin 1. So we're trying to mimic, right, that condition within these cells. And so what they're trying to gauge is the amount of the protein. So what we have here is a Western blot. All right, Western blot. Jamie, what's a Western blot? All right, a Western blot is a technique where you can look at, you're basically running an SDS page. So you learned that in biochemistry. You run a gel of protein through the SDS page gel, and you transfer the proteins from the gel to a membrane, a Western blot. And so you take an antibody that's specific for the protein to be able to see if it's there. So what we have is a control. So this is a control protein, and so that we can see if we actually see a real difference. And so this is the protein of interest, the ferroporin 1. And so we see that in the knockout, we don't have that the actual visualization of the protein, meaning that it has effectively been knocked out. All right, the next thing is to gauge the amount of iron that's accessible. And so they use two different methods. First, to measure the iron intracellularly, and then also to measure iron that's getting released, the extracellular. So the intracellular was done by the use of a turn-off iron fluorescent protein, so iron 2 probe, which is calcium. So it's a chelator that when it interacts with iron, it actually turns off. So what does that mean? Well, you have it being fluorescent, but when it comes into contact with the iron coordinates to it, it actually gets quenched, and so you get a turning off signal. So the more iron is present, the less fluorescence you're going to see. So what we see here in this image is the wild type, and then we have the knockout. We have a supplementation of iron, And what we see is that there is a less of a fluorescence signal, even though we're using the same amount of the calcine, we have less because there's less that's accessible in that cell. All right. And then in addition, they use a radioactive approach. They use an approach to measure radioactive iron 55 to measure the iron that's getting released from the cell. All right, so let's look at this, breaking it down. So we already saw the image that shows us that there's a decrease in the amount of iron that's available intracellularly, so less labile iron available. And so what we're seeing here, using, let's look at the left one here, a dosage of 5 micromolars of the hinokitiol with those cells, right? We're looking at, and supplementation with iron, we see that there is actually a release of the radioactive iron over time. We see that there's a trend towards it increasing. Okay, especially in the presence of the actual ligand. But then, and then you can compare that to wild type as well. But then on the right, we're focusing on the use of the calcine, which is that turn off probe, right? So we're essentially looking at what happens to its fluorescence over time as we're dosing different amount of the actual ligand. And so what you're seeing basically is a decrease in the fluorescence, and that decrease is a measure of like you having a loss of the amount of iron that's accessible. So what we're seeing is that the intracellular labile iron levels are decreasing in a concentration-dependent manner. All right. So then what they wanted to do was to measure also looking at the primary Kupffer cells. Because remember, these cells that we're looking at here were basically generated, right? But over here, they wanted to actually take out the Kupffer cells themselves from the flat iron mice, okay? And also from their wild type litter, or the wild type, just as a point of comparison. And so here, what we're looking at is the amount of intracellular iron and what happens in the presence of the actual ligand. And so what we're seeing is that we have in the ferroporin deficient, the mouse model that's representative of ferroporin disease, we have that increase in the amount of intracellular iron, but then with treatment of the ligand, you have that decrease in the amount of intracellular iron. Okay. So the next thing was to determine whether the ligand can induce hemoglobinization via a serum transferrin dependent manner. Okay. So the working model is that yes, it should be that the ligand will induce iron release from the liver macrophages. It will coordinate that metal and it will help deliver it to transferrin. But the question is that actually happening? So what they did was a series of experiments to try to gauge the iron interaction from the iron hinokitiol complex to the serum transferrin. So we're basically looking at this reaction here. I like to see reactions here. So what they did was they took serum transferrin and they dosed it with the iron bound to the ligand, okay, that complex, iron 3, up to 4 equivalents. And so what we're looking at here, we're not familiar with this, maybe not all of us, urea gel. Urea gel is a denaturation gel. So it's a measure of the relative stability of proteins. All right, so let's focus very quickly on what we have in this gel all the way to the left. The left is a representation of the relative stability of transferrin in the able form, no iron bound, versus iron bound. So if you think back to our discussion about iron binding and transferrin, I mentioned to you that iron binding stabilizes the protein. So we see the very same phenomena here via the urea gel, as we did with differential scanning calorimetry, where the further you move down this gel, the more stable you are, and the iron-bound transferrin actually moves further down relative to APO, and even relative to iron that's bound at one site of the protein. All right, so they use this as a way of following where the iron is going as they're dosing different mole equivalents. So they start with as low as 0.125 mole equivalent, all the way up to 4 equivalents. And they see after a certain number, so basically once you reach the level of 2, you basically have saturated the protein, okay? But before getting up to 2 equivalents, you have different populations of some amount of APO, some amount of single bound iron and some amount of hollow transferrin of completely saturated protein. All right. So this gel is actually able to help them to measure the relative population. I actually skipped over a lot of the details because I didn't think it was as important, other than to mention to you that as you would expect, this protein can bind 2 equivalents of iron. You do see that at 2 equivalents, that's where you get the actual saturation of that protein. Okay. That isn't always true of every metal complex. Some metal complexes have a lot more competition, but in this case, this ligand actually is able to effectively donate the metal to the protein at 2 equivalents. All right, so that has to do with the capacity to deliver the metal. So thermodynamically, this is in favor of iron bound to the protein. But in terms of the kinetics, how fast? Is this physiologically relevant at all? So they wanted to put this in a context. They said ferrokinetic studies in adult male humans show that serum iron trafficking to the bone marrow occurs within 10 minutes. So, and it peaks at about 4 hours. So we're talking about a time frame of 10 minutes. Is this exchange between the ligand and the protein faster than that time frame? Otherwise, this is just doesn't really make much sense at all. Is anything relevant? So what they did was an experiment to look at the to follow the kinetics, and they did an experiment where they did pseudo first order conditions. So thinking back all the way to general chemistry, we all talked about pseudo first order conditions. That means something's going to be in excess. So here, what's in excess is your protein. So I think we were taught 10 times is pseudo first order condition, but they claim 5 times is enough. Let's just go with that being okay. So you have the complex, you have 5 equivalents of protein or more, okay, because they go up to, I believe, maybe like, if it's 5 times 4, I think it's like 20 times more. And so you see this profile, they're looking at the observed rate constant and they're trying to fit this data. So taking all this into consideration, they determined that this exchange process within this concentration range is something that is within 75 seconds, something that's super fast, so physiologically relevant. So once that gets bound to the ligand, that ligand can rapidly deliver it to the protein, and now the protein can take its time to deliver it to the bone marrow and then generate the red blood cells. Okay. The next question was, all right, so when the ligand is iron bound, it can deliver the metal to the protein, but can the ligand, when it's not iron bound, can it interfere with iron that's bound to the protein, right? The opposite question. So this reaction here. And so they wanted to look at this, and they gauged this using the urea gel. So they did an experiment where they had iron-bound serum transferrin. They reacted with different dosages of the ligand, and they wanted to determine at what point do you start to affect the iron-bound to the protein. They obtained that at 165 micromolar, you start to get competitive. Okay, so the question is that concentration something that are you actually going to get that amount into your blood system? So they did a sort of pharmacokinetic dynamic experiment where they wanted to see the amount of that ligand that gets dosed in the circulatory system. First of all, how fast does it get cleared out, and at what concentration are you able to effectively get? So they did a number of studies. I'm going to just simplify it to just this key point, that they found that the ligand really rapidly gets eliminated from the circulatory system. It doesn't last long. And within 15 minutes after IP administration of that chronic amount of the 10 mg per kilograms, they get an effective concentration of 50 micromolar of that ligand present in the bloodstream with the amount of serum transferrin they have present. So the amount of serum transferrin is about 30 micromolar. So that amount should not, if you look at the data here, really should not have any impact on pulling iron from the protein. All right. So that talks about any intrusion with the with serum transferrin. So it definitely can deliver the iron to the protein, and the amounts that are present in the circulatory system should not affect how much is actually saturated in the protein. But the next question is, can the free ligand interfere with transferrin-dependent hemoglobinization in red blood cells? And so they're looking at red blood cell progenitors, meaning that things that can get transformed into red blood cells. So they focus on this cell, the murine erythroleukemia cells. Those can get transformed into red blood cells. And so what they observed was that the free ligand actually does not affect the amount of iron that's getting inside the male cells. So we can see that in this figure over here. where we're looking at dosing the cells with 0.165 micromolar of iron-bound serum transferrin and having different amounts of the ligand itself, we see that the same amount of iron is getting inside the actual cells. Okay, so that's not actually affecting the amount of iron that's getting in. And another thing is looking at the amount of hemoglobin that's getting produced, so the hemoglobinization. Are we seeing an effect there? And it turns out that the ligand does not affect the hemoglobinization. So that's what we observe on the right. Is not affecting it in the, so they basically dosed with something that is a globulin protein and also an antibody for the transferrin receptor. So the purpose of the antibody for the transferrin receptor is because they wanted to inhibit some amount of the transferrin going in. So that antibody is binding to the protein, it interferes with the transferrin that's bound to iron being interact with the protein. So they do see that the antibody decreases the amount of the amount of hemoglobinization that's occurring, but the presence of the ligand is having no effect whatsoever on this. The only thing that's affecting the amount of hemoglobinization is impeding iron transferrin getting inside the actual cells. Okay, and then they evaluated whether the iron transferrin that's generated from the actual compound, the iron bound to the ligand, can that induce hemoglobinization? So that's basically the same experiment that I was just showing you here, but this iron transferrin was actually made using the small molecule, so made in situ. When I say in situ, I mean transferrin, and you dose it with the molecule, and you have produced this complex. All right, and what they observed is the same trend, that you have the same uptake of the iron, and that's leading to hemoglobinization. So clearly, that iron transferrin that's formed from this complex does not affect this process. It actually is enabling the hemoglobinization. All right. Okay, so the next thing was inquiring whether that compound itself independently can induce the differentiation of the MEL cells into the red blood cells, because there was some thought that perhaps maybe the complex can enter into the cell via its own route that has nothing to do with transferrin. And so it turns out that that's not the case. That in another experiment that was done where it looked like that was happening, it appears that because you're typically using, you're doing your cell work and you're using media that has some amount of apotransferrin, that whatever amount of uptake that may have been happening was because of the transferrin that was present in solution. And they see something very similar in this experiment here, where you see the increase in the intracellular amount of iron when you dose it with iron transferrin, as you do when you dose it with the iron bound to the complex, and you see the impact of using the antibody for the transferrin receptor. You see how that affects the amount of iron getting bound. So it's clear that what's opposing the limits of how much iron is getting bound is iron that's bound to transfer, whether it's coming from iron already bound to the protein or coming from iron bound to the ligand that's being delivered to the protein itself. All right. So the very last thing was to examine using a clinically relevant system to determine whether there's a potential for this ligand to enable iron redistribution. So what did they do? So this is an ex vivo experiment. Ex vivo, what does that mean, Oscar? Ex vivo, what does that mean?
En persona, en person, en vivo es que lo está haciendo dentro del cuerpo. Yeah. Ellos tenían 3 mutaciones de 3 pacientes.
What's ex vivo? That's the question. What's ex vivo?
Because I think que tomaron las células directamente de una persona viva. Yeah, exactly. You're right.
That's what it means. And so what they did here was they isolated these monocytes. So these were immune cells that were made in the actual bone marrow. So they took it from bone marrow from 3 ferroporin disease patients. And so they had these mutations. So G80S, N174I, A77D. So these are representation of mild, medium, and severe ferroporin disease. I don't really know the details of this. I'm just going to buy with what they say. All right. So they worked with these monocytes and they wanted to see if they were differentiated into primary macrophages. They were able to observe that, but they were also able to observe the very same trends of how the ligand, okay, and here they're using the calcium fluorescence method, how the ligand with these different cells, right, how it has it can affect the amount of intracellular iron that's accessible in these particular macrophages, right? Because the idea that you want to move iron out of the macrophages and now it becomes accessible in the circulatory system. So they're able to see this from human cells, the monocytes that were transformed into the macrophages. All right, so I will note that there were a few things that were frustrating for me because they don't really explain some of the details of the animal work. They kind of just talk about what they did with then what kind of animals that they used, but they weren't very clear as to what were their wild types and how certain experiments were done. So it was a little bit frustrating to try to make sense of some of the actual experimental details. And there are a lot of, at least a couple of figures that were never actually discussed in the paper. So I'm not sure that was just an oversight, but for me, there was a little bit of trying to piece it together, obviously, and also looking at terminology. But I thought it was a pretty strong paper. All right, so the next thing I wanted to talk to you about, which is something we're going to talk about as we discuss further in the course, but it's important to bring this in the context of this idea of homeostatic control of metals. And so this is this idea of nutritional immunity, which is basically how your body responds to being infected by something that's pathogenic. And so the idea is that you basically want to either starve pathogens or, so I probably should retitle this terminology, or basically flood them with something that would be toxic to them. All right, so in nutritional immunity, basically that is a process by which a host organism employs molecular species to sequester essential elements in a defense mechanism against infection, but also can do the opposite to release essential metals. So let's go or release. All right, so here's this idea of how the human system can actually engage in a sort of defense, counter-defense mechanism with pathogens. So humans being the host, and then the pathogen being some sort of microbe that's entering. So you can have this idea of metal sequestration as what humans can do as their own defense. So it turns out that humans can employ peptides or proteins to increase the retention of essential metals like iron, and also to minimize export. So you can probably have sort of a suppression of, let's say, ferroporins, so that you don't have too much iron that's getting released from cells. So that would be how they can defend themselves by trying to minimize iron that, let's say, bacteria, as an example of a microbe, really thrive on iron. But then as a counter defense, you have a number of bacteria, as we already discussed, that have produced a large variety of small molecule chelators. So, for example, siderophores, or even very similar syncophores, which are obviously have an affinity for zinc. And so the idea is that they have a sort of a redundancy of these particular small molecules that have a very high metal affinity to be able to extract metal, iron, in this context of zinc, that's bound to a biomolecule with very high affinity. Okay, so that's how bacteria have evolved to try to fight back to be able to assess the metal. In addition, you also see a number of bacteria being able to use or express proteins that can recognize iron bound to biomolecules that are produced in the actual human system. So you have the example of being able to bind hemoglobin directly. You have the capacity of being able to bind the transferrin itself and take that metal and bring it into inside the cell environment, or even able to bind to the heme group itself. So you have the bacteria have evolved to be able to recognize these biomolecules that are present within our body. And this is a representation of within the actual cellular environment, how bacteria can actually mobilize ferritin, and does have storage of iron within the cells to be able to access that metal. All right, so something that's really important is this idea of metal levels and indicators of susceptibility towards infection. All right, so people suffering from iron overload diseases or requiring multiple blood transfusions, they would likely be at high risk for infection. Clearly because they have such an overdose of iron, that's something that pathogens would actually love. But then you have low metal levels in certain populations that may not in fact be something that would be considered an actual disease condition. That may be that the population has adapted to its geographical sort of endemic infectious diseases. And so they have sort of a naturally lower amount of metal than you would normally expect. So one of the key things is that the correlation of metal levels, let's say in blood, during a microbe infection may not actually warrant metal supplementation because it can be counterproductive and it can lead to just worsening of your clinical state. All right, so then this is another form of nutritional immunity where you have the process of a host organism employing molecular species, I meant to say release, not to, that's why I was trying to correct it before, to release essential members and defense mechanisms against infection. So as an example, and one of the major representation of this is copper itself. So copper is a super important metal, but many different organisms have a really high respect for it because it's something that can easily turn toxic. And so there is quite a bit of sensitive regulation, especially in humans and bacteria, which have less of a dependence on copper than humans do. And so you see that bacteria have a number of mechanisms employed to try to suppress the amount of copper that gets accumulated. So you have a lot of exporters for copper. So it turns out that humans actually release copper into their bloodstream as a way to try to poison pathogenic microbes. And so what you see as a counter defense is that you have an overexpression of these copper exporter proteins to try to minimize the poisoning that, potential of from the uptake of copper to basically avoid any of this retention of that metal itself. All right, so a number of this assessment of nutritional immunity has led to insights to potential new antibiotics. And so again, we'll talk about this later in the course. There's a big need for developing new antibiotic drugs, as our current drugs that are commonly used, they're basically reaching the end of their activity. So we have the potential of probiotics, and that is the use of bacterial strains that can be used possibly against highly pathogenic ones. As it turns out that bacteria produce their own antibiotics in a way to defend themselves against other more aggressive bacteria. You have people designing what are called these Trojan horse sideromycins. So what that means is that you're basically trying to synthetically alter siderophores, right? So that's something that bacteria will employ for binding iron. And you're making sort of a covalent bond with an antibiotic. So you're trying to trick bacteria to think that they basically are getting iron siderophore complex, but you're also adding in the moiety of the antibiotic that would then serve as a way to, to kill the bacteria. You also have efforts, more recent explorations of trying to target the suppression of copper export. So by targeting, I mean to try to prevent the suppression so that you have a lot more retention of copper within microbes. And even the idea of trying to develop copper ionophores that can effectively transport the copper, release it into bacteria. So these are a number of things that people have explored in part due to studies on nutritional immunity. Okay. So the other article that I had you read, you check your time, was this article here. And so this one has to do with metal regulation by a symbiotic relationship with the microbiome. Okay, so here, the gut microbiome is required for full protection against acute arsenic toxicity in mouse models. And so one of the things I want to start off with is just this representation of the microbes in our system. So more and more is appreciated that a lot of medical conditions are in fact due to a disruption of our microbiome, our individual microbiome. And so this gives us a little bit of perspective of things. I'm going to read some of these stats here. So microbiome in numbers. There are 100 trillion symbiotic microbes live in or on every person and make up the human microbiota. 95% of our microbiota is located in the gastrointestinal tract. 150 to 1, the genes in your microbiome outnumber the genes in our genome by about that ratio. All right, so you can go on and on. You can just see how much, the prevalence of microbes in our actual system. Okay. All right, so that being said, in this particular study, they wanted to assess the role of the microbiome in potentially regulating toxicity, in this case, the toxicity associated with arsenic. So let me just get a little bit of context. Since 2006, the US EPA has set the maximum contaminant level of arsenic in drinking water at a level of 10 micrograms per liter. Even though you can technically go down to 1 microgram per liter, but I guess in terms of finances, that's something that's more accessible. It's 10 micrograms per liter. It turns out that acute arsenic poisoning can lead to these different physiological effects: nausea, vomiting, abdominal pain, and severe diarrhea. And you can have also reports of this type of encephalopathy, which is a brain disease that can alter the brain function and structure, and also peripheral neuropathy. And so that when you have a chronic arsenic toxicity, so just longer exposure to to this particular element, then you can have what results into a multi-system disease condition. So what they did in this study was they used mice models to determine whether the human and also the mouse microbiome can exacerbate or potentially protect the host from acute arsenic toxicity. So that was something they wanted to assess. What role do the microbes have in these organisms in terms of potentially regulating toxicity of arsenic? And then they also wanted to examine the importance of this enzyme, this arsenic 3 methyltransferase, or abbreviated AS3MT, in terms of metabolizing arsenic. All right, so what was their approach? So they wanted to perturb the microbiome to measure how it affects the arsenic excretion and bioaccumulation. They wanted to determine whether that enzyme and microbiome increase survival and thus play a protective role. So this was something very important to gauge. And then to determine whether human microbiome transferred to mice, so basically taking a human sample, putting it into an animal, can provide host protection. And they also wanted to see whether there was any donor being from the human to the animal, dependent variation against protection. So what were the mouse models they used in order to assess this? So they use, or if not necessarily mouse models, but the type of animal treatments. So animals that was treated with antibiotics, so they use this broad spectrum cephalosporin as their antibiotic. They also use transgenic mice. So that is genetic material that's transferred from another organism. They use germ-free animals. So that's basically, they don't have any microbes. And then they use a gnotobiotic, which they have only certain strains of bacteria. All right, so these are the different animals that they use for this particular study. All right, so what was the first thing? Perturb the microbiome to measure how it affects arsenic excretion and bioaccumulation. All right, so perturb the microbiome. So in this context, then they're using that broad spectrum antibiotic as a way of perturbing it. So a dosage that's enough to affect the levels of the microbiome. So they use this mice, the C57BL6, which is the black mice, and they were either administered the broad spectrum antibiotic stuff, or they did a sham treatment. Sham basically is you're using your buffer solution, your background solution that doesn't actually contain the antibiotic. So they did this for 2 days prior to and throughout a 14-day exposure to 25 and 100 ppm inorganic sodium arsenic. So this is the form of arsenic, arsenic 5 in drinking water. All right. So what they were assessing was the content of iron in the mice stool and also in the organs to get that picture of how much is accumulating in the body, so the biodistribution, but also how much is getting released into the being excreted. All right, so here's the data. So maybe a little bit hard to see here. So what are we looking at here? So we're looking at treatment of arsenic at 25 ppm and 100 ppm. Okay, and we're seeing in the white dots are the sham delivery, and the red dots are the broad spectrum antibiotic, the SAF. All right, so here we're looking at the amount of arsenic in the stool. So the sham versus the SAF. All right, so Alex, so how much arsenic is present? Do you see a difference in the 25 ppm versus the 100 ppm? All right, you see a difference, and you also see a dose-dependent difference, right? So you see here, we're leveling off at about 20 ppm, that's in terms of exposure, But over here, you're seeing something that's leveling off at 100 ppm, basically in line. Yeah, that's fine. In line with the amounts it's actually getting administered, right? But you also see a difference that has to do with the actual treatments. So the sham, no antibiotic, and then the one with the broad spectrum one, you see that we basically are affecting the amount that's actually getting excreted, right? So it's not getting excreted. We don't have movement of the arsenic through that. So where is it going? So this is giving us a perspective of where potentially the arsenic is going. We have, again, the 25 ppm versus 100 ppm, and we're looking at the distribution in different tissue: liver, spleen, heart, and lung. Okay, so Jaylene, okay, what do you see here? So we have, again, the antibiotic versus the white, which is no antibiotic. What do we see in terms of the distribution? Right. Right, so we see it particularly in the lung. We see quite a bit of an accumulation, and it becomes even more pronounced when you go into the 100 ppm. Here now we see more in the actual liver as well, okay? So we have less excreted, but more being distributed into the body, which is bad, right? All right, so then the next thing was to determine, that sounds bad. The next thing was to determine whether the transferase, this enzyme, right, it converts arsenic, and the microbiome can increase survival and thus play a protective role. So what they did was they took this enzyme, which facilitates the methylation of arsenic. Okay, so basically transforms inorganic arsenic into, let's say, a type of organic arsenic, and thus can biotransform that metal. So that's that element. And then they also wanted to examine a form of knockout mice. So basically knocking out the enzyme. So Brenda's not here for me to ask what does knockout mean. But anyways, knock out that enzyme and see what effect it has. So it turns out that these knockout mice are known to experience greater arsenic-induced cytotoxicity because they don't have the capacity to biotransform the arsenic itself. These mice were examined to determine whether they would have greater sensitivity to arsenic treatment compared to wild type. And then they also wanted to examine survival of the mice populations when exposed to 10, 25, and 100 ppm arsenic. All right, so these are the treatments. The knockout mice are treated with the SUF and the sham, and then the germ-free knockout mice, so they don't have any microbes in this study. All right, so this is what we're looking at, 25 and 100, okay? And then what we're focusing on is percent survival over time, okay? In the presence of sham, let me write this bigger, and SUF. Okay. All right, so Oscar, let's look at the 25 ppm. And you're looking at survival in the sham over the 20 days of exposure. What's happening to the mice? Survive. 100% survival. Okay, none have died. But then when you have the SAF treatment, okay, so this the introduction of the antibiotic, now we're starting to see loss of life, right? We have at 25, we have about 40% survival. Okay, so the treatment with the antibiotic is having a significant role, but even the knockout mice without the antibiotic still are surviving. So what do we see at 100 ppm? All right, so Brenda, since you're back, 100 ppm, we're looking at survival. Are the mice dying without the treatment of the antibiotic? Just the sham. Are they surviving or are they dying? They're dying. And you can see that by day, let's say 28, you have no survival, okay? And then you have this getting even more affected by the treatment with the antibiotic. You have survival decreasing earlier. Okay. All right. And then what do we see at the bottom? So at the bottom, we're looking at treatment with 10, 25, and 100 with knockout and the germ-free knockouts. Okay, so now these organisms, in addition to the germ-free ones, in addition to not having the enzyme, they also don't have any of the germs. And what we observe is that, especially at 100 ppm, the germ-free knockout mice are dying even earlier on. And you get 100% death by what seems to be 5 days, right? So there definitely seems to be a correlation between the the actual germs, and to some extent, the importance of the enzyme itself. I make a point here that it would have been nice to see sort of a germ-free wild type of avian existed as a comparison to what we see with the knockout mice. All right. So they also wanted to get, I'm going to go through this one pretty quickly. They also wanted to see, well, is the molecule itself somehow affecting mortality in a way that's independent? And it turns out that no, the actual molecule itself is not increasing mortality by itself. All right, so the next thing was to determine whether the human microbiome transferred to mice can provide host protection. All right, so this was done by basically taking some human excrement and then making it into a slurry, and then basically providing by oral gavage to the mice. So that basically is putting it inside them. All right, and so what we're looking at here is the germ-free knockout mice receiving this transplant prior to the treatment with the arsenic. Okay. And so they call this particular line of animals the humanized F0 because they received this material from humans. Then any animal that was birthed from these animals were considered the F1. So these are the humanized from birth knockout mice. All right. So they wanted to see the variability of mice survival based on the different donors. All right. So let's try to make sense of this, what we're seeing here. So we're looking at percent survival, okay? And then we're looking at different conditions. So let's try to, there's a lot going on here. So we're looking at the knockout mice under the condition of sham, so not treated with any antibiotic. And we see that we have up to about, let's say, 27 days, some amount of survival, but at that point, all the animals have died, okay? But then we're also looking at the F0 and the F1 mice. So these have been supplemented with human material, and we see a phenomenon that's pretty equivalent in terms of the survivability. There's some variation, but at the end of the day, pretty equivalent. So this is with 100 ppm treatment with the arsenic. And then finally, the mice that are germ-free, so they don't have any microbes, you see that they die earlier on. The next thing was to assess, well, general population, like different donors, how is that affecting survivability? And you can see, depending on the donors, I'm not gonna go too specific, that survivability ranges. We have like down to 22 days up to like almost 40 days. So depending on where the estimate came from, we have different amounts of effects. So it's obviously very dependent on the microbiome of the individual. Okay, then finally, they were looking at identification of microbiome members that correlate with survival. So they wanted to identify, are there any specific microbes that are leading to the survival of the animals in the presence of dosages of the arsenic? So what they did was a 16S rRNA gene sequencing on the stool samples from humanized mice at the onset of the arsenic exposure and also at day 7 prior to mortality. So what they did was a taxonomic classification by blasting the data set against the ribosomal database project. So that was their way of being able to try to identify particular microbes. So they try to determine the gut bacteria that exhibit detoxification properties with survival modeling of the 100 most abundant operational taxonomic units, or OTUs. All right, so here we have a layout. These are all the different microbes that were examined. These are from the different donors and the different mice and the donors they received from. And you have this heat map that basically gives you the amounts of the corresponding microbes that are present. So the black bars indicate the strains that were observed on both day 0 and also day 7. So again, other microbes that were identified, the heat maps that correspond to those microbes. And so an important note as far as looking at the x-axis is that a negative coefficient indicates a protective association, while positive coefficients are associated with an increased hazard. So from this assessment, right, from the overall study, they identified this particular bacteria, the fecal bacterium. And so they wanted to study the potential for that bacteria to serve a protective role. So according to this data set, it does seem to be protective, right? The association is, but is that actually physiologically relevant? All right, so what they did was they did a combination of the previous studies and also the known promotion of this microbe as a probiotic. So basically, they wanted to see the bacteria's ability to produce this enzyme, led the research team to hypothesize that it would provide protection against the arsenic toxicity. So let's look at this. It turns out that they were not able to colonize that bacteria by itself in the mice, so they had to do a co-colonization. And so they had to co-colonize it with E. coli. So even though bacteria can be pretty stable in the in vivo environment, oftentimes when you try to isolate them or work alone, they're very susceptible. So in this context, they had to work with it in the presence of E. coli. So what was actually observed here? So we have the assessment of survival with just the E. coli by itself, mice that are germ-free, so no microbes, and then the mice that have been supplemented with the E. coli plus this other microbe. And so what we observed here is that total survivability was more prevalent, even though all the animals still died in the presence of that microbe. So it suggests that it does have the capacity of serving a protective role, or at least extending life when treated with 25 ppm of the arsenic. So it seems that there is a correlation from that study that was done, that this microbe may be leading to it, and that might suggest that some of the other microbes here are also contributing to it, but they didn't actually characterize those other microbes. Okay, any questions or doubts? Okay, that's very scientific.
No, because it's very difficult quantifier rating the último.
Profesor,
Sí.
la cantidad de organismos que ellos trabajan, ¿no? O sea, tienen en unos 4 individuos, eso no afecta a la gente.
I would think so, and you would think that maybe they would try like one donor over maybe a few different animals to see a similar trend. It may be just limitations of how much material and how many animals they have accessible. That's oftentimes what limits a lot of these in vivo studies. Other questions? Oscar, why did you like the other paper more? Scientifically.
Scientifically, because it's more, for me, it's more complex, and when it reading, I understand everything with the conclusion, with the result that present. With this, I feel, I don't know, maybe I might be bad, but I feel there's need a little more information, because only you see the viability for the treatment, but I think that's just enough for one conclusion specific.
Yeah, I agree with you. And just like Jaylen was saying, maybe we needed more animals to assess different conditions. Yeah, because even early on, we never saw a wild type control to compare to knockout to see that is an actual true correlation with the loss of that enzyme. So yeah, I would agree with you. What about you? Anyone? Doubts, questions?
Like the study doesn't really come to a full conclusion because of so many missing factors and the fact that there are trillions
Yeah.
of microbes in the system, not any possibly like the ones that is one of them.
Yeah.
And the paper says that the only use microbiome might be food, but what about humans that have that problem?
Yeah, exactly.
You don't have this problem with people with IBD and other,
Yeah.
another bowel disease, how is that affected? And I feel like that's just, they're missing a big scope.
Yeah, of course. And very clearly, like when you have now a combination of different organisms, how is that going to impact the physiological factor? It's very true. All right, so yeah, let's end here. And then we don't have class on Thursday, so we'll resume next Tuesday with a seminar from your fellow classmates. All right, I will send the next assignment tonight. All right, bye, y'all.
Goodbye.